The central nervous system is organized structurally and functionally into gray matter, which contains cell bodies, and white matter, which contains our myelinated and unmyelinated axons. We'll now zoom into three specific structures: the spinal cord, cerebral cortex, and cerebellar cortex. In cross section, the spinal cord has a butter shaped pattern of gray matter, represented here as the darker blue, and this kind of H connecting the two, surrounded by white matter along the other outside. The arrangement will reflect the functional organization of both motor and sensory pathways. The ventral horn of the gray matter is going to contain motor neuron cell bodies. The dorsal or posterior horn contains our sensory axons and our interneurons. Our sensory neuron cell bodies are located outside the spinal cord in the dorsal root ganglia. In the cerebral cortex, it consists of 6 layers and contains two main neuron types: pyramidal cells and granule cells. This multi-layered organization enables a complex integration as well as enables our higher cognitive functions. And the cerebral cortex is this gray matter that's on the outer surface of our brain. Our cerebellar cortex is also located on the outer surface of the cerebellum. We can see this here, and this is where the cerebellum is located. The cerebellar cortex has 3 layers: a molecular layer, a Purkinje layer, and a granule cell layer. The Purkinje neurons are large and very distinctive and are the primary output cell of the cerebellar cortex, which is really critical for motor coordination and knowing where your body is in space. And this is kind of the breakdown of these white matter and gray matter properties. Our peripheral nervous system includes peripheral nerves and ganglia, which connect back to the central nervous system, and they connect the CNS to limbs and organs. There are sensory and motor signals that travel through these pathways. So some sensory receptors, they are located in places like our skin. They help us to detect environmental stimuli like pain, temperature, touch. And some examples include free nerve endings. This picture here are our Pacinian corpuscles, our Messner's corpuscles, and then Ruffini corpuscles. If we broke them down, free nerve endings are really here for pain and temperature. Messner's are light touch. Pacinians are vibration, and Ruffini is stretch. The dorsal root ganglion contains, as I showed you before, connected kind of to the spinal cord. This contains cell bodies of our afferent sensory neurons, and these are pseudo-unipolar neurons. They're surrounded by satellite cells. Unlike our autonomic ganglia, the dorsal root ganglia does not have any synapses within the ganglion itself, just the neuron cell body. The neuron cell body kind of has this body here, a single attachment, and then its axon going in both directions. The neuromuscular junction we learned is our specialized synapse between a motor neuron and a skeletal muscle fiber. One motor neuron and the fibers that innervate it form a single motor unit. The peripheral nerves contain thousands of axons and they're all bundled together. There are 3 connective tissue layers that organize them: the epineurium, which is the outer dense connective tissue, dense irregular connective tissue lining that surrounds the whole nerve. We have perineurium, which is the dense connective tissue that surrounds the fascicles. And then around each individual axon, there is endoneurium, and this is loose connective tissue. Nerves can be afferent or sensory or efferent motor fibers. This is looking again at our peripheral nerve, thousands of axons. This is kind of your deep dive into the histology of the nervous system. I recognize there's so much more we could layer on in depth, but for now, I just want you to have recognition of these structures and be able to start to identify their different components and how they look under histologic stain. This is a review of kind of our peripheral nervous system. And if you really like these types of images, these come from Med Boot Camp. They have a lot of really great resources for studying for your USMLE Step 1 board exam or your NPLEX step one board exam, kind of your biomedical sciences. So they are a great resource, and that's where these kind of breakdowns are pulled from.